I'm very happy to introduce my colleague, Dr. Pengyuan Yang. Pengyuan got his PhD from the second military medical university in 2004 and did his first postdoc in the same university in Dr. Xue Tao's lab. In 2009, he joined Dr. Xiaofan Wang's lab at Duke University. And on 2014, Pengyuan became a principal investigator in Institute of Biophysics, Chinese Academy of Sciences. Pengyuan did several very interesting works in the past few years. I guess he will present one of the study today. So let's give the time to Pengyuan. The title of his talk is HBV-initialed carcinogenesis and immune microenvironment. Okay, so Pengyuan, you can start.
Okay, so thanks for Jizhong's introduction, and thanks so much, Wu Chuan and Xiaolei's invitation. It's my great honor to be here today to introduce an old-fashioned topic, the hepatitis B virus-initiated carcinogenesis and the immune microenvironment. So firstly, introduce the etiology of the HCC. So it's hepatocellular carcinoma. Hepatocellular carcinoma accounts for about 90% of primary liver cancers and ranks as the fourth most prevalent cancer worldwide. As you can see, okay, sorry. You can see here that the HCC primarily develops from the hepatitis C virus infection, also has other, such as the echo use or non-echo fatty liver disease in North America and Europe. However, in the East Asia and Africa, hepatitis B virus infection is the predominant etiological factor. Although Chinese government initiated a universal HBV immunization program in 1992, however, the majority of HBV infection occurs at early age with very persistence. So the patients are still very young. So it's still a major etiological factor in China. So in China, there has 383 people die from liver cancer per year in China. It contributes 91% of deaths from liver cancer worldwide. In China, it's about 80% of the HCC cases in China are attributable to HBV. So even Professor Wu Mengcao in corresponding reports published in Hepatology report noted that China has invested to distance itself from its reputation as the great country of liver cancer. So how serious? So you can say, although from the annual report of China Cancer Registry, so liver cancer ranks the second cause of cancer death in male and the third in female. You can see it's important that so liver cancer is the most common. It's the most commonly diagnosed cancer and the leading cause of cancer death in men under 60. So recently, several groups utilize the single-cell RNA sequencing technology to investigate the pathology of HCC based on the huge patient resources in China. So you can see Fanjia group reported the distinct phenotype of CD8 T cells in early relapse HCC ecosystem. And Zhang Zheming group published several decent articles on dynamic properties of immune cells in different tissues of HCC patients and identified several immune cells subsets during the progression of the liver carcinogenesis. That they also identified the LAMP3 positive disease in the HBV positive liver cancer tissues. So the different etiology, whether they have different mechanisms. So in contrast to a majority of cancer types, so the transformation of the liver in HBV-associated HCC often follows from accompanies long-term symptoms of chronic hepatitis, inflammation, and cirrhosis. Besides adding this transformation, HBV play a crucial role in modulating the combination and activation of both cellular components of the tumor microenvironment and some intrinsic factors of carcinogenesis. So in this talk, I'm going to introduce 3 parts of our studies. So including the HBV-initiated tumor microenvironment, so the HBV-specific immune recognition, and the interactions of the HBV with host immune system. So firstly, I introduce the story on the tumor microenvironment. So, oh, sorry, I have a. So this is the worldwide well-known Barcelona clinical liver cancer staging system. You can see because liver is a tolerance organ, the patients are often diagnosed at the advanced stage. Sorafenib is the very few present new line therapeutic agent. However, the efficacy is not so satisfactory. So clinical doctors, including Dr. Wu Mengcao, still recommend surgical resections in China. So what's the problem? So in 1917, we found a published paper that published in the Journal of Clinical Oncology reported the improvement of the overall survival in patients who are HBV negative. However, in the HBV single positive patients, there is no evidence of this improvement in the overall survival, which means that the different etiology contribute to the different effect of the drug treatment. So this report really touched me since this is the majority type of liver cancer in China. But unfortunately, the present first-line medicine doesn't have any effect to improve the prognosis. So we try to identify some mechanism of this drug resistance. Maybe this disadvantage happen in the microenvironment from the different etiology factor. We firstly repeat the preclinical studies in nude mice. Actually, the effect is very good. You can see the treatment of Sorafenib. I'm sorry?
怎么了？
However, once treat these experiments, when we repeat these experiments in immune-competent black 6 mice, the treatment seems not so good. Even the low dosage of the Sorafenib slightly increase some of the tumor growth. So this data indicate that lymphocytes may mediate the Sorafenib resistance. So to investigate the possible mechanism, we employed a qPCR-based assay on the profile of cytokines and the chemokines. Of this panel, we can see the CCL22 and CCL17, both ligands to their receptor CCR4, were the top significantly upregulated gene upon the Sorafenib treatment. Consistent with my previous research, CCL22 was selectively elevated in HBV positive tissue, tumor tissues, and could be upregulated by HBV infection. So about actually it's an old story. So over 10 years ago, I have identified a HBV-initiated pathway that TGF-beta regulate the microRNA-mediated CCR22 induction and promote Treg cell recruitment via its receptor CCR4. So in this study, we identified that Sorafenib treatment accelerate the CCR22 production via TNF-alpha and the RIP1 kinase activation pathway in HBV-positive microenvironment. The antagonist of CCR4 could rescue this phenotype of the drug resistance and inhibit Treg cell infiltration. So if this phenotype is organ specific, so recently we found some answer. In the same cytokine array, we found another cytokine, L8, was significantly induced by HBV infection. So this induction was regulated by HBX and HBX-induced ERK activation. So L8 is just a very common cytokine, but unfortunately, it doesn't exist in mouse. So therefore, so when we explore the functionality of the IL-8 in mouse model, so we have to explore a human 6cell1 transgenic mouse is the receptor to investigate the function in vascular metastasis. So we can see the data. I'm sorry, the computer. Okay, so we can see the activation of the L8-6R1 cascade by overexpression of the IL-8, that they dramatically increase the metastasis in liver. However, in the same time, it doesn't significantly consistent, but this increment is not consistent in lung metastasis. So this data suggests the L8-6R1 cascade selectively enhance intrahepatic vascular metastasis. So negatively, so the L8 selectively induce the gap latent TGF-beta in liver sinusoidal endothelial cells. So we identified the 6cell1 interact with the gap, the protein in the membrane of the liver sinusoidal endothelial cells to enhance the TGF-beta expression. So this local TGF-beta activation that is in the liver, that specifically in this organ could enhance and provoke the prefunctional regulatory T cell proliferation to suppress the anti-tumor immunity. So this finding revealed a hepatitis B associated L8-6R1 signal axis that mediates the local immune escape of HCC to induce the intrahepatic metastasis. Okay, so in this part, so we initiate some story under HBV positive microenvironment that including the HBV infection could produce some variproteins such as HBX and HBS, that they can induce some cytokines such as TGF-beta and L8 and RIP1 kinase activation that induce the immune escape and drug resistance. And this kind of the microenvironment accelerate the the linkage of the inflammation from the inflammation to carcinogenesis and metastasis. So my next question is, how immune system could recognize these tumor antigens since at a very component, because it suppose have some tags on this kind of the tumors. It's a viral initiate carcinogenesis. So maybe the immune system could recognize some tags of the various components. So to develop this and investigate the T-cell immune recognition and the downstream of TCR, so we set up a new technique termed Ti-ATAC-seg to identify the identify the single-cell epigenomic and T-cell profiles. So at the beginning time that we were going to determine the single-cell TCR-seg, some students in my lab, attendant students, that reminded me that we should do some additional determination on the nuclear, because the nuclears are going to discard it. So then we use this Ti-ATAC-seg that established that allow us to determine the single-cell TCR, the protein markers, and the corresponding epigenetic changes. So as shown in this final panel, you can see that we already identified several TCR pairing sequences in COVID covalence individuals that already recovered from the COVID-19, and the corresponding transcription factors activation to each different TCR receptor. And importantly, with this powerful technique, that we can at the same time monitor each cell, especially the CD8 T cells memory signal pathway, that whether the Tbet activation or the F4 BAF activation, to whether how the intracellular signal pathway was activated in each T cells. So how about the HBV positive HCC? Firstly, we determine the overall epigenetic profiles by ATAC-seg. You can see the tumor infiltrating T cells exhibit a continuous spectrum of epigenetic profiles in the downstream of TCR signals that involved in T cell activation and exhaustion. So with this Ti-ATAC-seg, we identified the single cell epigenetic changes in response to each TCR receptor, in which we separated them into 7 clusters. And these clusters were identified to the different cell type and different activation stage. And for this marker of the TCR, okay, so the TCR clone types, we found many TCR clone types that were shared in these patients. Importantly, by using a junket reporter system, we identified several TCRs that are HBV specific, that can be induced by the HBV peptide pools. So, since this kind of TCR are HBV specific, they can recognize the HBV-induced tumor cells. But what's happening in these T cells, since we already know that this kind of the tumor tissues are immune suppressive, actually, they have lots of Tregs, and the CD8 are not functional. So armed with this Ti-ATAC-seg, we are able to monitor these T cells clone migration and distribution in different tissues. We can see the T cell expansion capacity was higher in peritumor than that in the tumor tissues. So these tumor infiltrating lymphocytes are under exhausting, actually. So how it was under exhausting? So we further next examined the epigenetic signatures of these T cell clones. In this panel, we can see the enrichment of the SMAD3 transcription factors in HBV-related CD8 positive T cells indicate that the effector function of these cells might be suppressed by slow TGF-beta signals. Our data also provide some additional interesting clusters that some of the, such as ERF1 and DRON-D activated the or dysregulated T cells. Some of them are stem-like, some of them are effector and or a memory phenotype. So, in this part, we set up the single-cell Ti-ATAC-seg of HBV to determine the HBV positive HCC that revealed a dynamic or heterogeneous T cell response against HBV-derived antigens. More robust expansion of the virus-specific T cells was identified, and they correlate with the prognosis of these patients. So then, the next question is, where the antigen come from? Since in the has already carcinogenesis, so it's the viral titers are very low, so they don't have much replications. So, but still has the HBV DNAs in the nucleus. So the HBV virus is very small. It's very small virus. So, however, it can produce several types of the HBV DNAs. So it's including the CCC DNA is circulated, circulated closed circular DNA. So it has several dozens copy in the human nucleus, and the integrated DNA. That this kind of the intranuclear DNAs are considered to be the primary contributors to the development and prognosis of HBV-associated liver disease. Moreover, the HBV DNA can integrate into the genome in the infected liver cells that couldn't be get rid of, which progressively also contributes to the hepatic carcinogenesis. So in the early time of the 1987 and recently the 2016, so some articles have reported the hotspots of the HBV integration sites, but the oncogenetic function are still unclear. So particularly, so how the HBV DNA long-term persistent within the human nucleus and contributes to the malignant of the liver disease, it remains unclear. Yet how the HBV, especially how the HBV DNA communicate with the human chromatin. So I need to thank my collaborator Ji Xiong and his friend Hu Jiazhi that they helped me to explore the technique of 3C GTS, a highly sensitive method to identify the regions interacting with the HBV genome after the cross-link digest and then ligase and remove the cross-link and make some PCR, LAMP PCR to amplify the regions and do the sequencing. We use two kinds of the HBV cellular models, including HBV infected, the HepG2 NTCP cells, and the HBV integrated, the cell line HepAD38, that this kind of two cell lines were employed in this 3C STGS analysis. And also we found two the integration site in the AD38 cells. So consistent with the previous report, the HBV DNA interact with the whole host genome. That's not surprising, and strongly correlates with active histone modification. So interestingly, our data indicated the HBV host DNA context are significantly enriched in the enhancers that are associated with the H3K4 methylation 1 and be regulated by the host gene KMT2C and 2D. And also we found, sorry, I missed this slide. So we found that the KMT, the host gene KMT2C and 2D can regulate the expression of the HBV virus. So in addition, the integrated HBV DNA organizes the chromatin loop. You can see, how about the integrated HBV DNA? That this kind of DNA is organize the chromatin loop structures. You can see that linking active host DNA adjacent to the integration sites. So this kind of the integration may be very functional, although it couldn't regulate the virus replication. However, it can regulate the host genes. So in this part, our study provided a regulatory landscape of spatial organization of the HBV DNA and the integrated DNA with the human genome, which serves as the molecular basis for shared epigenetic machineries between HBV virus and the host genome. So actually, we are still working here to, we are going to identify the cccDNA and the integrated DNA, whether they are related with the immune system. So this study, I need to thank my graduate students Yu Mao Jun, Yang Bo, Zhang Changlu, and my assistants Gao Yanan. For the technique, I need to thank the Chen Rensen Lab and Stanford McDavis and Chen Liang, Beijing University Ji Xiong, and Li Wenhui, Huang Ailong. For the clinical samples, I need to thank the National Liver Cancer Center and the Chinese PRO General Hospital and the Beijing Union Medical College that they provide the clinical samples. Thanks so much for your listening, and I'm ready for the questions. Thanks.
Okay, thanks Pengyuan for the talk. So any questions from the audience?
Hi, Pengyuan. This is a really beautiful talk. So I think I have like two questions. So one is general, one is specific. So maybe I don't know much about this like virus field. So just in general, so between the infection of HBV and the like detectable level of like liver carcinogenesis, I guess there are a few. It's a long pass, I guess, because
yes.
the HBV you can have like a very chronic infection. So is there any critical point along this path that you can either like keep the genome integrated, or like it causes like the irregulated like cell proliferation? Or is there any marker we can like use to determine these like critical point?
Oh yes, Shirley, you really asked a very good question. Yeah. Yes, because the mechanism of this kind of the integration are not well identified. So there have several hypotheses of this kind of the integration. So actually, we are going to establish some mechanistic study to do this. Yeah, and we firstly we use the 3CSTS a similar technology, we can identify because the yeast cells are different. So we are using the single cell determination to identify the integration site in yeast cells and identify the transcriptions in yeast cells, whether they can regulate the host genes. And another issue we are going to do is we use the transgenic mice to explore the different integration, whether has some different functions in the mouse carcinogenesis. But this is very challenging because it don't have the liver infection, because the mouse couldn't get infection by HBV. So this is a really problem in our system. So
I see.
Yeah, we tried some experiments to monitor, to mimic this kind of the infection. Yeah, but it's still not working very well. So yeah.
So maybe a related question is like, so the liver cells, a lot of them are polyploidy cells. That doesn't really like proliferate. Is there any like difference between the like HBV's preference for the ploidy of the cells when it's infected?
Yeah, because if we culture the primary hepatocytes, it is multiploidy. So we can see that, and this kind of the hepatocytes need to be fully differentiated. Otherwise, it couldn't get infection, get a good infection.
So is that because of the expression of the receptor that's for the HBV?
Yeah, probably. But maybe some other co-factors. Yeah, the Wenhui has identified the NTCP. It's a very important receptor. Yeah, the expression levels really downregulated in the co-culture system.
Yes.
Cell culture system. Yeah, it's important, but maybe some other factors that also mediate this kind of infection. Yeah.
Do you think there will be a de-differentiation like before the
yeah.
Because the polyploid cell doesn't really like to undergo cell cycle. If I understand correctly.
Yeah, probably. Yeah, I think you give us a very good suggestion to to investigate the multiploidy. Yeah, it's some kind of the difficult for us to to do some mechanism sequencing. But I think this is important. Yeah.
I see. Okay, so the other question I have is, so the HBV-induced IL-8, so that's through the ERK pathway, right?
Oh yes. The HBX, yeah.
Okay, so but the ERK can be activated by like a plethora of receptors like EGFR or TCR. Like a lot of receptors can activate ERK, but not necessarily further activate downstream IL-8. So is there any other like cofactor or co-pathway you think here is play a role here?
Yeah, that's a very interesting point. Yeah, we identified the SBX can activate ERK. So yeah, ERK is very common and not so specific for this cytokine expression. Yeah. But the IL-8 is a very important cytokine for the metastasis. It's already identified. But in this study, we identified the local mechanism that it's only happen in the hepatic metastasis. So mainly promote the intrahepatic metastasis, such as some organ-specific mechanism.
I see.
Yeah, because the only liver have the sinusoidal endothelial cells, and all the other vessels that they doesn't express the GAP protein. So that's the point. Actually, we identified some other cytokines such as IL-18 that are also published in cancer research. The IL-18 can regulate the T17 cells and regulate the immune escape of the microenvironment. Yeah, that's complicated.
Okay.
Yeah.
That's good. Okay. Okay, maybe one last small question. Is there any physiological role of the IL-8 in the hepatocytes?
Oh, yeah, very possible, I think. Since the CXL1, okay, there has some clinical investigations. So that is the receptor CXCR1 are not highly expressed in the tumor tissues. Actually, such as some of the immune cells, that they're supposed to express the CXR1, but once they were infiltrated in the tumor tissues, the CXR1 expression was downregulated. So, so we speculate that the CXR1 mainly functional in the endothelial cells. Yeah, that's our point to use this model to identify this.
Okay, great. Yeah, thanks so much. Really beautiful work.
Thank you so much.
Okay, more questions?
Hey, Pengyuan.
Hi, Chen.
I have, I maybe missed it, but my question is, you're saying that IL-8 and CXCR1 can induce endothelial TGF-beta release, right?
Yeah.
So I guess my question is how you know the dominant role to induce Treg cells in the liver is through this endothelial TGF-beta, but not the other sources. So you know, IL-8 can CXCR1 access also can induce other cell to generate TGF-beta, not only the endothelial cells. I guess my question is how you know this endothelial generated TGF-beta is the main source to induce Treg cells.
Yeah, that's a good point. That's yeah, you are expert on the TGF-beta signal pathway. Yeah, so yeah, so we just exclude a pathway that mediate this function, but we believe that tumor cells can express very high levels of the TGF-beta. Yeah, actually in the liver, the fibrosis are also reported to regulate TGF-beta. Yeah. Just think that the CXCR1 mediated the endothelial activation also participate in the tumor tissues. So maybe this kind of endothelial cells can make some function, yeah, in regulate the immune escape. So that's why, yeah, we elucidate the function of the endothelial cells. Yeah, yeah, so, it's definitely true that other cells is.
So
yeah,
so the other thing is this this signal from the IL-8 and the CXCR1 is induce, you know, transcription of TGF-beta level, or just to promote the release of the TGF-beta from endothelial cells. Do you know?
Probably, yeah. We determined the latent TGF-beta that are not released to the cyprinate or some blood. So it's just a local membrane expression. Yeah, they didn't release. Maybe we didn't determine that even. Yeah, because the latent, the membrane TGF-beta, we think it may be more important for the local immune microenvironment. Yeah. So
have you tested the RNA level of TGF-beta in the endothelial cells after you stimulate them with IL-8?
It should be, yeah, but we didn't because it has already reported the gap that stabilized the protein expression. Yeah. So the gap signals couldn't regulate the mRNA levels. Yeah, so it's stabilized the protein in the membrane. So that's our story on this.
I see. So it's not really transcription regulation signal. It's more post-translational to release the latent TGF-beta from the endothelial cells.
Yeah.
I see. Okay. Okay. Thank you.
Yeah. Thank you.
Okay. Any more questions? Okay, so I guess no further questions. So then let's thank Pengyuan again for his wonderful talk. Thank you, Pengyuan.
Thanks for the questions.
Thanks so much, Pengyuan.